but thank you for the opportunity to present today. I'm going to be presenting on some of the work I did in Bert Klumperman's lab last year under my postdoc, and that's regarding the synthesis of some double hydrophilic block copolymers that are based on SMA type copolymers, and specifically for the synthesis of divalent cation tolerant SMALPs. So Alice has already touched on some limitations of the SMA type copolymers, but there's a couple of factors that are necessary for successful solubilization in most cases. One of the major factors being appropriate amphiphilicity of the copolymer, and this is achieved in a number of ways where you can have variable comonomer composition, where this can be SMA 2000 with a 2:1 styrene, or you can go to alternating SMA 1:1, which we know doesn't work so great. To get around the struggles of making a RAF synthesized SMA 2000, there are a couple different strategies you can employ, such as using alternative comonomers to styrene or maleic acid. So some examples of that have been the employment of tert-butylstyrene as opposed to styrene, or acrylic acid as opposed to maleic acid. And alternatively, you can employ post-polymerization modification of your maleic anhydride units, as has been demonstrated by Gestal's polymers, the BZAM series, where you have a certain partial modification that improves the hydrophobicity of the copolymer. Another important factor for your copolymers is an appropriate charge distribution along the backbone. So you want to maintain water solubility of your copolymer so that you have sufficient water solubility and electrostatic repulsion between your SMALPs. And we know that with traditional SMA, you have a sort of limited pH range dependent on maleic acid's pKa. So you can't really have soluble copolymers below about a pH of 3 to 4. And also you have chelation of your maleic acid units with divalent cations. So, this is the general concentration range you're looking at for SMA type copolymers, where generally SMA to DIPMA, it varies a little bit within this range, but you generally have very poor tolerance to divalent cations, particularly magnesium and calcium, where exceeding a certain concentration of the cation or decreasing the pH too far yields complete protonation of your backbone or chelation to your divalent cations, resulting in a collapse conformation and eventually precipitation of your copolymer from solution. Now, our strategy to try and overcome these limitations is to actually employ SMA within the context of a double hydrophilic block copolymer, so a slightly different architecture to the generally employed SMA copolymer, where the addition of the hydrophilic block attached to the SMA should facilitate self-assembly of the copolymers into aggregate type structures, micelles or vesicles, as opposed to precipitation of the copolymer. Now, double hydrophilic block copolymers are generally composed of two water-soluble blocks, where one of the blocks is permanently water-soluble, so in this case, the blue block, and the other block is water-soluble under ambient normal aqueous conditions, and upon application of a stimulus, in the case of SMA being a divalent cation or a proton, that yields a loss of charge in that particular block of polymer, and that yields collapse of one of the blocks, which under appropriate conditions, high enough concentration of the cation, high enough concentration of the copolymer, etc., you should get aggregation. And specifically in the case of the divalent cations chelating to the SMA block, the aggregation of those block copolymers then results in HPICs or hybrid polyionic complexes. So the idea is that in the context of synthesizing SMALPs with these block copolymers, not only do we add the sort of improved tolerance to divalent cations and hopefully pH, but you also add the steric stabilization in addition to your electrostatic stabilization of the copolymer for the SMALP. So the polymers of interest in our particular study are just normal alternating SMA as a starting point, which we know isn't ideal for solubilization of membrane proteins, but it provides a good starting point to assess the solution properties of these block copolymers. We then went with a slightly more hydrophobic analog of SMA, an alternating tert-butyl SMA type block copolymer, where the permanently hydrophilic block, the permanently water-soluble block, is poly-N-acrylomorpholine. So the approach or the synthetic strategy we applied for the synthesis of these block copolymers is, of course, to use Raft polymerization for the synthesis of the first SMA block. So it can either, if you're looking at the black structure, it's just styrene alpha-malate anhydride, or if we include the red tert-butyl group, that is then our tert-butyl SMA block. This then can be chain extended in organic media to yield our block copolymer. And this is, of course, an amphiphilic block copolymer that we need to then treat with basic conditions to afford the double hydrophilic block copolymer. We can also approach the synthetic strategy in a slightly different way, and that would be to take our SMA macrochain transfer agent, ring open that into our water-soluble macrochain transfer agent, which we just applied for the one copolymer, and then this can be chain extended in aqueous media to also afford the double hydrophilic block copolymer. And once we're at this stage, and we have a polymer that's very happy in water, we can go on to characterize its solution properties, how pH tolerant, how divalent cation tolerant is it, and then apply it in the solubilization of DMPC vesicles as a proof of concept to see if we can make SMALPs under very salty conditions, essentially. So some results for our block copolymer synthesis. For the synthesis of just our macrochain transfer agents, we have very quantitative monomer conversions and very nice low dispersities with targeted molecular weights. And these were then used subsequently in a chain extension experiment indicated by 2, where we made 2 amphiphilic block copolymers chain extended with our permanently water-soluble block. Again, quantitative monomer conversions and very nice low dispersities. So we have a copolymer sample with very similar chain lengths and composition, which is exactly what we want for very well-defined solution properties. The alternative synthetic strategy, where we ring opened our macrochain transfer agent first and then chain extended in aqueous conditions, also proceeded with high monomer conversion, but slightly high dispersity, which I'll discuss in the next 2 slides. So once you have your amphiphilic block copolymer, hydrolysis of the copolymer is quite facile, and it's actually a much nicer hydrolysis procedure compared to the hydrolysis of just SMA type copolymers. As you know, when you add these copolymers to a basic solution, immediately the polymer precipitates until it is fully hydrolyzed. In the case of block copolymers, the attachment of the permanently hydrophilic block actually prevents precipitation of the polymer, yielding good surface contact of the anhydride units with the alkaline conditions, the alkaline media, resulting in a very rapid hydrolysis under quite mild conditions. Within half an hour, you have your fully hydrolyzed polymer, which you can then purify. And we just showed that you get complete hydrolysis with FTIR. So we characterize the block copolymers with size exclusion chromatography and DOSI NMR. And we basically just confirmed with this that we did indeed have a block copolymer. So the fact that our protons for our respective block copolymers have similar diffusion coefficients means that we very lightly successfully made our block copolymer and not just a mixture of the 2 homopolymers. And size exclusion chromatography showed something similar, where we had a shift in our molecular weight distribution to high molecular weights. And for the second synthetic strategy, where we chain extend our macrochain transfer agent in water, we saw slightly different results, where we seem to have a mixture of our block copolymer and the starting macrochain transfer agent. And we assumed that was because of cleavage of our Raft N group during the hydrolysis of our macrochain transfer agent, making this a suboptimal synthetic procedure for very nice, well-defined block copolymers. So we then moved on to characterize the solution properties of our block copolymers. I'm going to start off with the SMA, alternating SMA block with the acrylomorpholine block. So we expose this either to decreasing pH. Sorry, I think this is cut off at the bottom. This is in concentration of divalent cation and pH. So we exposed our block copolymers at two different concentrations. Firstly, at a low concentration of 3 milligrams per mole, increasing up to 50 milligrams per mole. We exposed that from 0 to 200 millimolar of calcium. On this plot, this is magnesium in the middle. And we use HCl for the protonation of our copolymers. And we have a couple of different ways in which the cations can interact. We can have intermolecular chelation or intramolecular chelation. And the efficiency of this chelation also depends on the pH a little bit. But despite that, we saw some very different results for the SMA PNAM copolymer. So in terms of the calcium sensitivity, we can see at 3 milligrams per mole. This is, by the way, this is just SMA. So this is not the block copolymer. This is just the SMA copolymer. As soon as you start increasing the calcium, you can see immediately this crashes out of solution by a DLS analysis. But the block copolymers, at 3 milligrams per mole, you can see as you start increasing the calcium, it essentially increases slightly in hydrodynamic diameter, which is self-assembly into micelles, and increasing to 50 milligrams per mole increases this tolerance way more. And you can see up to 200 millimolar, you essentially still have your copolymer in solution. For magnesium, for 50 milligrams per mole of polymer, we saw very good tolerance. But as you decrease the amount of polymer, you can see that the tolerance to magnesium decreases incrementally, but still very good tolerance compared to just traditional SMA. Unfortunately, the block copolymer did not have improved tolerance to a decreasing pH, and that is because essentially your hydrophilic block, which is essentially supposed to keep your block copolymer in solution, this hydrogen bonds with your protonated acid groups, and you end up just forming a precipitate. So it actually decreased the tolerance of our block copolymer to a decrease in pH. And this analysis was repeated for our third-butyl SMA block copolymers, and we saw almost the opposite tolerance, which was quite interesting. So for calcium, we saw very sort of slight tolerance to calcium, still improved compared to just normal third-butyl SMA copolymer, but this sample above a certain concentration of calcium actually formed a gel. But it was very nicely tolerant to magnesium. So this block copolymer up to 200 millimolar magnesium was able to form micelles. And again, a decrease in pH did not result in or this block copolymer did not have improved tolerance to a decrease in pH. So what's actually more helpful is to represent the charge ratios. Instead of looking at concentration of magnesium or calcium, we rather represented this data as a ratio of charges between our magnesium or calcium and our polymer. So essentially below a Z ratio of 1, you don't have equivalent number of charges and you should have your polymer in solution. And above a Z ratio of 1, you should have fully screened all of your charges. And generally this would result in precipitation for the SMA or third-butyl SMA copolymers and self-assembly or precipitation for the block copolymers. And as you can see for the SMA acrylomorpholine block copolymers, you have improved tolerance when you apply higher concentrations of polymer because it evens out your Z ratio. But again, you can see that this particular polymer is a bit less tolerant to magnesium, more tolerant to calcium, and exactly the opposite is observed for the third-butyl SMA block copolymer. And we hypothesize this is due to a combination of factors. So for starters, your calcium cations are slightly larger with higher coordination numbers or higher preferred coordination numbers. And the block copolymer PSMA PNAM is actually more hydrophilic. So in this case, the combination of this cation with the more hydrophilic block copolymer results in micelle formation. And on the other hand, magnesium being a smaller cation preferring lower coordination numbers, this only results in micelle formation with the less hydrophilic and more sterically bulky tert-butyl SMA block copolymer. So depending on the amphiphilicity and the steric bulk of your SMA component of the block copolymer, you can have different tolerances depending on the identity of the cation, which we found very interesting. So to characterize the extent of micelle formation in a little bit more detail, we did some DLS and NMR analysis, where essentially we showed that for our SMA acrylomorpholine block copolymer, we had barely any micelle formation up to very high Z ratios, and this is essentially very high magnesium concentrations up to 200 millimolar. For the SMA block copolymer treated with calcium, we had quite successful micellization, but not complete, up to very high calcium concentrations of 200 millimolar. And then for our tert-butyl SMA acrylomorpholine block copolymer, again, we had complete micelle formation at slightly lower Z ratios, but this corresponds to 100 millimolar magnesium. And we characterize what type of morphology aggregates we were creating with treating our block copolymers. These are concentrations of between 100 and 200 millimolar of either magnesium or calcium. For our SMA acrylomorpholine block copolymer, you can see even up to high concentrations of magnesium, we have these diffuse structures, essentially because we did have free chains and not aggregates as micelles. For calcium, you can see from DLS that we actually have more micelles than we have free chains. And from TEM, we could see some spherical morphologies, which would suggest we have some sort of diffuse micellar structures. For tert-butyl SMA, for both magnesium and calcium, we saw these very well-defined spherical structures, which would indicate that we do have micelle formation. But in all of these cases, the copolymer maintains full water solubility. We then moved on to see if we could apply these block copolymers at these higher cation concentrations for DMPC solubilization. And starting with the tert-butyl SMA block copolymer, we could see that slightly increasing the concentration of magnesium up to 8 millimolar resulted in efficient solubilization of DMPC vesicles. But as soon as we increase to 32 or 100 millimolar magnesium, you see the solubilization slows down quite significantly. Given enough time, you can see that the optical density does drop, and DLS does show us that in all cases, we are making structures of around 9 nanometers. And when analyzing these structures with TEM, you can see we do have the formation of these sort of 14 to 17 nanometer discoidal structures with similar morphology to that of traditional SMALPs. So in the cases of the 32 millimolar magnesium and 100 millimolar, our previous analysis has showed that this sample predominantly constitutes micelles, but the acrylomorpholine block itself does not interact with phospholipid bilayers. So we hypothesize that there is some equilibrium between free chains and micelles, where depletion of free chains into the solubilization essentially pushes this equilibrium back from micelles to free chains, allowing for a slower but ultimately successful solubilization. We did repeat this for the calcium series, but as I said before, this copolymer becomes a gel after 16 millimolar calcium, and so we could only really show that it can be employed for solubilization up to 8 millimolar, which is pretty much still within the applied range for just tert-butyl SMA copolymers. We repeated this analysis for SMA PNAM copolymers with both magnesium and calcium, and alternating SMA is generally known to be pretty poor at solubilizing DMPC vesicles, and we can see that here in the DLS data at, the DLS data at 0 millimolar. So we have slight disruption of our vesicles, but it's definitely not a successful solubilization. But as we start increasing the amount of magnesium or calcium in some respect, we get a shift to smaller hydrodynamic diameters, which would suggest we have successful solubilization. And analyzing these structures with TEM also shows that in some cases, for example, at 8 millimolar magnesium for alternating SMA, we actually have a successful formation of SMALPs, as well as at 8 millimolar calcium. But we also successfully formed the SMALPs at 100 millimolar magnesium. But in the case of 100 millimolar calcium, we actually generally just have the formation of polymer-based micelles and almost no solubilization. So in this case, we're likely forming very stable micelles that actually can't, the shift in equilibrium back towards free chains is just not efficient enough to yield good solubilization of our DMPC vesicles. And this has actually shown a really nice method to tuning the amphiphilicity of your alternating SMA in such a way that you get successful solubilization, but without the need for post-polymerization modification of your copolymer, which is very nice. So conclusions, we were able to make two different types of block copolymers, two different chemical compositions, and with very low dispersity and targeted molecular weight, which is great. And these copolymers were amenable to self-assembly under certain conditions, up to very high concentrations of our divalent cations, which is great. The pH tolerance was not improved. However, because of the hydrogen bonding water-soluble block, so that can potentially be improved by using a hydrophilic block that does not necessarily partake in hydrogen bonding, although that is quite a tall order. Another nice thing about these block copolymers is essentially you retain your raft N group at this chain end, and that can be aminolized to make a thiol, or you can do a Sumi reaction and insert a biotin moiety, which we've already successfully done in our lab. So these block copolymers can also then be nicely employed in surface immobilization procedures. And we also showed that these block copolymers under these crazy concentrations of divalent cations can actually successfully interact with phospholipid bilayers, creating SMELPs that are both electrostatically and sterically stabilized. And if you have modified your chain end, these can then be immobilized for subsequent analyses.
Thank you very much, Lauren.
I would like to thank the following people for your contributions and funding. Thank you so much.
Thank you.
Running a little behind. What do you think, Michael? Should we have the break?
Just one question from the audience. If anybody has a question for Lauren, please. If not, I'm wondering what the biocompatibility and toxicity profile is of your hydrophilic block and the diblock copolymers. Have you looked into that?
So acrylomorpholine, polyacrylomorpholine is actually considered one of the best replacements to PEG in the biomedical industry. So it has really good non-immunogenicity, it's non-cytotoxic, it's quite biocompatible, it's supposed to prevent protein absorption as well. So it is actually already employed in quite a few areas in the biomedical industry. So it's perfectly well, I wouldn't say perfectly in all cases, but it's quite biocompatible. The SMA, not so much, but in our case, that's not really an issue. We're trying to disrupt the membrane. But yes, it doesn't decrease the biocompatibility of the copolymer.
Great. Is it approved for use in vivo, the hydrophilic block?
I think so. I'm not too sure. I don't know if it's up there with PEG yet in terms of being FDA approved, but I would have to double check that. But I think it is. It's been employed in quite a few studies already. So I would have to double check.